Hello. Today we're going to dive into the bubbly world of microbes, specifically the tiny yeast that powers our beer, your bread, and maybe even helps set your mood. Let's raise a glass to the fungus among us. Our bodies are home to trillions of microbes, bacteria, fungi, and even viruses. Many of them are friendly roommates. They help us digest food, make vitamins, and keep germs out. But when that balance tips slightly, it causes chaos, specifically in our gut, which affects inflammation and our mood. Early on, we figured out that microbes can do amazing things. They transform milk into cheese, dough into bread, and then grain into beer. That brew tank is basically a bubbly little ecosystem full of working microbes living their best lives. Brewmasters act like micromanagers, balancing temperature, oxygen, and sugar to keep yeast happy. But if wild bacteria enters, like lactobacillus or pedococcus sneak in, they can sour the batch. Now, funny thing, those are the same bacteria that make sour beers and kombucha on purpose, so sometimes a mistake can taste delicious. Meet Saccharomyces cerevisiae, the yeast behind beer and bread. This is what we call a facultative anaerobe, which means it can ferment with or without oxygen. Its cousin, S. pasteurinalis, makes those delicious crisp lagers. Beyond beer production, yeast also helps scientists study genetics and disease, so not bad for a single-celled fungus. So next time you sip a pint or enjoy that first bite of that warm, tasty bread, remember you're tasting the teamwork between humans and microbes. Every beer is a tiny science experiment that happens to taste amazing. So on my birthday, I'll raise a glass or three to the yeast that keeps life delicious and discoveries bubbling. So cheers to those microbes.
Hey guys, today we're going to be learning about the magical koji. Now, if you haven't heard of koji before, it is a mold, Aspergillus oryzae, that grows on a substrate and is used in other fermented products to make familiarly known as soy sauce, miso, sake, and it produces that umami flavor that we're all familiar with. Hopefully you are. So, how is koji made? It's a four-step process. You take a grain or soybean and you let them soak overnight. Then you steam them through until they are cooked and you want them to cool at room temperature. You definitely don't want them too hot because your mold will not grow. So we inoculate a koji starter, or you can self-grow it from corn husks and incubate that at 27 to 35 degrees Celsius for 24 hours. So we've successfully inoculated our koji and it's time to add it to some other ingredients because koji itself cannot is not typically eaten on its own. It's usually added to other ingredients to add a depth to the ferment and also enzymatic activity, which we'll look deeper into, is very important in fermentation. So you're going to take the inoculated soybeans and mash them up and add it to a salt brine for our soy sauce to create a moromi, which is left to age in a container that doesn't have to be airtight. And that ferments for about 3 years, 1 to 3 years. And to stop the fermentation process, pasteurization does need to happen because you don't want the microbes to continue creating flavors when you've already achieved the flavor you need, if that makes sense. So, what are the enzymes doing in Aspergillus oryzae? How are they surviving? And what is it that is adding to this umami flavor or creating it? So, koji is a two-step fermentation process. There's our aerobic solid state of fermentation, which we did see with inoculating our substrate with the Aspergillus oryzae. And that mold is providing a lot of enzymes, the main star being protease. That protease is breaking down those proteins into our smaller amino acids. And those amino acids do contribute to the umami flavor. There's also amylases hydrolyzing the starches in the substrate and breaking them down into sugars, which is something that we need in our second step of fermentation, which is our anaerobic liquid state fermentation, and that's when we added the brine. We also add lactic acid bacteria and yeast to that because those guys are going to add to our fermentation even more, and the sugars from the amylase are going to provide fuel for them. So while the enzymes are working away, they're taking the fuel in, their byproducts are creating these flavors that we know very well in soy sauce. So if we take a closer look at the sporulation of our Aspergillus oryzae, you'll notice that they have elongated condyle chains, which make up these long spores that arise from the substrate, which you can see with just with the naked eye. But if we look a little closer, they have been found to have 8 chromosomes and encode proteins for more than 100 amino acid residues. And their genome information has not been well known until recently. So some of these things we are continuing to discover when it comes to the Aspergillus oryzae. So like I was saying before, the proteases play a main role in our flavor for what we're looking for in the amino acids. Each of these individual amino acids that are released interact differently with flavor compounds. So we're going to focus on the glutamate today, which when it's released, it interacts with our brain's umami center and instantly gives that recognizable flavor. Another one would be alanine, which is released and interacts with the sensory organs and the tongue, and that also contributes to the umami flavor. Another thing I want to touch on is what glutamate does for our other microorganisms and the symbiotic relationship that they hold. The glutamate provides a nitrogen source for the lactic acid bacteria and the yeast. The nitrogen source is used as an intermediate of the citric acid cycle in these microorganisms that are anaerobic. So them being able to accept the nitrogen that is being released from the proteins, thanks to our protease, then we are able to continue that fermentation process, and they're able to continue living their little microorganism lives. So as we can see, koji really is a magical source of fermentation. The fact that it can just be added to any other ferment and its enzymatic activity is driving these microorganisms to do more. It really is incredible what's happening from the small scale to the larger one. And, without these enzymes, we wouldn't be able to taste many of these delicious flavors. Well, thank you for joining me on our tour of koji today, and you and I were both able to experience the not only rich history, but the rich flavors of koji and soy sauce.
I selected the food option for the homework assignment, and we'll be discussing lactic acid fermentation and some of the organisms responsible for turning cabbage into sauerkraut. Humans have been fermenting food products for thousands of years. While vegetables did not leave traces of evidence for later discovery, the first evidence of kimchi exists in iconography from around 2000 BC, so almost 4,000 years prior to van Leeuwenhoek's discovery of bacteria. Even though the science behind the process was not understood, cultures were able to use fermentation to preserve food. Coincidentally, I'd started experimenting with fermentation at home just a few months ago, and the process of making sauerkraut couldn't be more straightforward. Shredded cabbage is combined with salt. I've experimented with salt ratios anywhere from 2.5% to 3.5% by weight. The cabbage and salt is massaged or pounded, which causes the cabbage to release moisture that will form the brine. This mixture is then placed in a covered container to ferment. There are many lactic acid bacteria at work, but I'll briefly talk about a couple. First is Leuconostoc mesenteroides. It's active early in the process when heterofermentation is occurring. During this stage, glucose and fructose are metabolized to produce lactic acid, CO2, and a small amount of acetic acid. As a result, the pH drops and L. mesenteroides makes way for Lactobacillus plantarum, which is active during the homofermentative process. Again, glucose and fructose are metabolized. However, the sole product of homofermentation is lactic acid. The fermentation process lowers the pH, inhibits organisms that would cause spoilage, and gives the sauerkraut its defining sour flavor. The lactic acid bacteria are facultative anaerobes and occur naturally on the cabbage, so the use of a starter culture is not necessary.
Hi, everyone. My name is Joseph Martini, and today I am going to be talking about kombucha. Kombucha is a fermented tea, and one of the microbes that make that possible is going to be the Comagactibacter xylanus, or K. xylanus for short. Kombucha begins with tea, water, and sugar, but through microbial teamwork, it becomes a tangy and slightly fizzy drink that is rich in organic acids and probiotics. This transformation happens inside what is called the SCOBY. SCOBY stands for the symbiotic culture of bacteria and yeast. Now, the SCOBY is not just a blob on top of the tea, it's an actual living biofilm created by the K. xylanus. And so at the beginning, yeast, like Saccharomyces, break down sugar into ethanol and carbon dioxide. Then K. xylanus oxidizes the ethanol into acetic and gluconic acids. This is what gives kombucha its sour flavor. It also protects it from contamination. Now, what makes K. xylanus unique is its ability to produce what is called a bacterial cellulose, and this forms the structure of the SCOBY. The cellulose acts as a floating mat where bacteria and yeast coexist. So the bacteria are going to be near the top because they are aerobic and they need oxygen. And so at the top of the tea is going to be that oxygen-rich surface. And yeast are going to be deeper below where the sugar concentration is going to be higher. It's basically a perfectly balanced microbial ecosystem. Using glucose from the tea, the K. xylanus then converts some of it into energy, but the rest is converted into something called the UDP glucose. And this is what weaves the cellulose fibers to kind of give the SCOBY its structure. And I was actually able to find the genes within the K. xylanus that do this. It's going to be BCSA and BCSB. Now, the acids that are produced lower the pH, which means that it can add flavor and it can keep unwanted microbes out. What's left is a lightly carbonated probiotic tea created entirely through microbial metabolism. And at the end, Comagactibacter xylanus does not just ferment kombucha, it builds a very structure that allows fermentation to happen. It turns simple sweet tea into a self-sustaining living system. And this is a perfect example of how microbes transform basic ingredients into something complex, stable, and delicious. Thank you for listening.
Hello, today I'm going to be talking about the microorganism Gluconeacidobacter azotocaptans and its relation with coffee plants. Coffee is well loved by many, including me, and what many people may not realize is that microorganisms play a huge role in helping these plants grow, especially in nutrient-poor tropical soils. The bacteria Gluconeacidobacter azotocaptans forms a beautiful partnership with coffee plants that helps it grow and also reduces the need for chemical fertilizers. This relationship is a perfect example of how microbes can support sustainable food production. This coffee-loving bacteria is a gram-negative, rod-shaped bacterium that belongs to the family Acetobacteres. This family includes many bacteria known for converting sugars into acids or alcohols. Azotocaptans can be found in the roots and stems of tropical plants, including coffee plants and sugarcane. This microorganism should be cultured on nitrogen-free media to encourage it to use its nitrogen-fixing abilities. And even though this microbe needs oxygen, it still thrives in low oxygen areas like those plant tissues within coffee plants. The key process that makes this bacterium so valuable is called biological nitrogen fixation. Essentially, the bacterium converts nitrogen gas from the atmosphere into ammonia. Coffee plants can use the ammonia to build up essential molecules like proteins and chlorophyll. This is a pretty energy-intensive process, but the coffee plant provides the sugars the bacterium needs to fuel it. The partnership allows for both organisms to benefit. From an agricultural perspective, this relationship has huge benefits. By naturally enriching the soil with nitrogen, azotocaptans reduces the need for synthetic fertilizers. That means lower costs for farmers, less environmental pollution, and healthier soil ecosystems.
Fermentation is credited to Louis Pasteur's germ theory. There's many different types of fermentation, including alcohol fermentation, symbiotic fermentation, and lactic acid fermentation, which is what we'll take a closer look at today. Specifically with cucumbers, which are said to have originated in India, but have been seen all over the world across many different cultures for thousands of years. The fermentation of cucumbers into pickles are called pickles in the United States, Europe, and Canada, but have many different names across different cultures. Cucumbers have a very short shelf life due to an enzyme on their skin that softens them. This makes them the perfect candidate for fermentation. For industrial fermentation, a 5% solution of sodium chloride is added to the cucumbers to pickle them. But for most at-home pickling, a 2% solution is used. That way the lactic acid bacteria can still grow and kill off any harmful pathogens without making the pickles too salty. Lactobacillus plantarum is a specific lactic acid bacteria on pickles. They're gram-positive, non-motile, non-spore-forming, and they're rod-shaped. They're also facultative anaerobes, meaning that they can survive in aerobic or anaerobic conditions. They also prefer a more acidic environment and colder temperatures. They undergo lactic acid fermentation by first going through glycolysis, breaking their glucose down into 2 pyruvates, releasing 2 NADH molecules. Then this pyruvate is catalyzed by lactic dehydrogenase coded by an LDHL gene to turn it into lactic acid. This lactic acid is then used as a waste product. This process is continued until this lactic acid builds up in the brine, making the brine more acidic. Lactobacillus plantarum are resilient enough to survive the acidity of our GI tract and actually contribute to our microflora. This acidic condition that the pickles are in contribute to its sourness, their crunchiness, and the saltiness of the brine. Anything else is just an added flavor of the pickles.
Have you ever wondered why ginger beer tastes tangy and is naturally fizzy? I'll tell you a secret. It's powered by microbes. Ginger beer is a fermented drink. We mix water, fresh grated ginger, and sugar, and allow that to sit in a covered, but not airtight, jar for 3 days. After 3 days, foam and bubbles should appear at the surface. It's alive! The star is Lactobacillus, a lactic acid bacterium. With partner yeast, it eats sugar and makes lactic acid for tang and carbon dioxide for fizz. Yeast add a bit of ethanol and aroma. Many lactobacillus are heterofermenters. Their pathway, the phosphoketolase, turns one sugar into lactate and CO2 and ethanol, or acetate. That's the biochemistry behind the flavor we love. As fermentation runs, pH drops, so it becomes a little more sour, and CO2 rises, meaning more sparkle, and the flavors build. If it's unpasteurized, you keep some of the live microbes. Pasteurizing keeps flavor but removes most viable cells. Quick thought. If you double the sugar, what changes first? Sweetness or carbonation? Well, the answer is more fuel for microbes means more CO2 and acid, which can actually taste less sweet. From 18th century brewers to today's kitchens, ginger beer is living chemistry in a glass, thanks to Lactobacillus.
Hello, and welcome to Microbiology 234 with a short presentation about Streptococcus mutans. Streptococcus mutans is a gram-positive, aerotolerant, facultative anaerobic bacterium belonging to a group of mutans streptococci that are found in the mouth, living mostly on the surface of the teeth within the biofilm. Although S. mutans is a facultative anaerobe, meaning it can live with or without oxygen, it prefers an anaerobic environment where it thrives on fermentation in very low levels of oxygen, the mouth being the ideal environment for this bacteria to stay in homeostasis. It primarily metabolizes carbohydrates through glycolysis, which produces lactic acid. Its evolution has been significantly shaped by human dietary changes. It is believed to have acquired the genes responsible for producing enzymes that act like molecular glue to attach a glucose sugar to other molecules, such as proteins, lipids, or other sugars. This allowed it to form robust biofilms, or dental plaque, using sucrose through horizontal gene transfer from other lactic acid bacteria like Lactobacillus. This evolutionary event likely coincided with the onset of human agriculture and the increased consumption of carbohydrates. In today's time, sugar sources are even more abundant, making it challenging to control the population growth of S. mutans. S. mutans, also known as sugar bugs, survives by metabolizing carbohydrates, but thrives in an environment rich in glucose, hence sugar and soda, all the fun treats. Biofilms, or dental plaque, form from the release of lactic acid during the metabolism of carbohydrates, creating areas of tooth decay. S. mutans is a prevalent bacteria living in the mouth that can cause harmful damage to the teeth, leading to other health risks such as gum and heart disease. Fortunately, the bacteria can be managed by decreasing sugar intake and staying consistent with flossing and brushing. Thanks for listening to this brief review on Streptococcus mutans. Have a good day.
The human body is a remarkably rich environment for the flourishing of diverse microorganisms. The presence of so many different species on and in our bodies reminds us that we are more ecosystems than we are individuals. When our microbial communities are disturbed, either via antibiotics, environmental toxins, or the foods we eat, severe health issues can occur. Let's zoom in on the small intestine, where we find heavy digestive secretions and bile salts, rich nutrient availability, and a short transit time relative to other major microbiotic regions of the gut. We also find the many different organisms that live here, in all three domains of life. Together, these microbes have been shown to ferment carbohydrates, synthesize and modify vitamins important for human host cells, and boost the human immune system. When Giardia enters the system, the lower intestinal microbiome can be greatly disturbed. Giardia has two major stages in its life cycles. During the cyst stage, the protist is protected from environmental factors and can survive in cold water or harsh conditions for many months. Once the cyst reaches the stomach of the host, acids and enzymes work together to break down the cyst walls, releasing 2 trophozoites per cyst once it reaches the small intestine. During the trophozoite stage, the flagellated, double-nucleated, pear-shaped trophozoites attach to the mucus layer by using a ventral adhesive disc. This weakens the mucus layer, dramatically changing the composition and diversity of the microbiome by removing biofilm and competing with other anaerobes. This process is what causes the malabsorption of nutrients and further symptoms in more severe infections. Most evidence agrees that the presence of Giardia is correlated with a lower diversity of microbiota, and thus a less healthy gut. However, it's possible that the presence of Giardia actually strengthens the gut against further, more serious parasites, which means it might be more of our friend than we have historically thought. While the harmful impacts of infections are commonly known, there remain many opportunities to further study the mechanisms of microbial disruption, non-antibiotic solutions, and potential protective effects of Giardia for overall human health.
Are you the proud owner of a passageway to life called the vagina? Or perhaps someone in your life has a vagina you care about? A key aspect of vaginal health is the dynamic balance between different microorganisms that most commonly reside there, including many strains of lactobacilli, which act as a defense system against harmful bacteria that also reside in this sensitive zone, such as various types of candida. The most common extremely beneficial microorganisms found in the vaginal microbiome include Lactobacillus rhamnosus, Lactobacillus reuteri, Lactobacillus crispatus, Lactobacillus gasseri, and Lactobacillus jensenii. All of these bacteria are rod-shaped, gram-positive, facultatively anaerobic, and have been shown in studies to help reduce yeast populations by making it difficult for the pathogens to attach to the epithelial cells of the vagina. When the pH balance shifts within the vagina, these good bacteria can die off, making it much more likely that a woman could develop vulvovaginal candidiasis, more commonly known as a yeast infection. Many factors can disrupt the pH balance of a woman's microbiome, such as hormone changes throughout her menstrual cycle or life, staying in wet swimsuits, wearing non-cotton underwear or tight clothing, and even using tampons or irritating soaps and sprays can harm the delicate balance of the microbiomes there. Extended exposure to pools or hot tubs with harsh chemicals can also contribute to an inhospitable environment for those lactobacilli, killing off the good bacteria in the vagina, which can greatly increase a woman's risk of infection. I hope that understanding these crucial components of a healthy symbiotic vaginal microbiota can help 3 out of 4 women that suffer from infection-causing pathogens. Thank you for your time.
Hi everyone, today I'll be summarizing my project on how the human microbiota influences health and disease. The human microbiota is the collection of microorganisms that live in and inside us, including bacteria, fungi, and viruses that form complex interactive communities. In a healthy state, these microbes protect us, help digest food, train our immune systems, and even produce vitamins. But when this balance is disrupted, known as dysbiosis, the same helpful residents can turn opportunistic and cause disease. Several factors can open what's called an opportunistic window. These include immunosuppression, antibiotics, stress, aging, and even introducing microbes to places they don't belong. For instance, E. coli is harmless in the colon but can cause urinary tract infection when it enters into the urethra. Likewise, emotional stress or sudden dietary changes can shift microbial populations, creating opportunities for infection. Antibiotics are a big contributor. They wipe out both harmful and beneficial species, leaving space for overgrowths like yeast infections. Maintained balanced microbiota across all body regions is essential, and while I think the oral microbiome is especially important, every region deserves care and respect for its healthy resident microbes. Zooming in on a specific ecosystem, I chose the male reproductive microbiome, particularly the foreskin. This region contains diverse microbes and immune cells like Langerhans cells, which can either protect against or promote inflammation if overwhelmed. The most abundant genera here include Corynebacterium, Staphylococcus, Anerococcus, Prevotella, and Winkia. Interestingly, circumcision can alter this environment, reducing anaerobic bacteria, changing how the tissues interact with moisture and oxygen, which may influence susceptibility to infections like HIV. From this community, I focused on Corynebacterium glucuronolidicum, a gram-positive facultative anaerobe commonly linked to chronic bacterial prostatitis. It feeds on shed epithelial and prosthetic cells, and while usually overlooked, it can become aggressive when conditions favor its growth. This organism responds to antibiotics such as quinolones, tetracyclines, and azithromycin, though resistance has been observed. Understanding its behavior could help detect and treat genitourinary infections earlier, highlighting the importance of microbial balance and precise clinical sampling. Thank you.
You probably know by now that your body is home to trillions of tiny organisms like bacteria and fungi that actually help keep you alive. These microbes live all over us, on our skin, in our gut, and along our mucous membranes. They help digest food, make vitamins like biotin and vitamin K, and protect us by taking up the space and nutrients that harmful microbes would otherwise use. But like any ecosystem, balance is everything. When we take antibiotics, use harsh soaps, or even deal with stress and poor diet, we can throw off this balance, a condition called dysbiosis. When that happens, harmful bacteria can grow unchecked, leading to inflammation, infection, or even diseases like colorectal cancer. So keeping our microbiota balanced isn't just nice, it's essential. One very consequential microbial community is the vaginal microbiome. It's warm, moist, low in oxygen, and slightly acidic, perfect conditions for the Lactobacillus species to thrive. These good bacteria turn sugars into lactic acid and hydrogen peroxide, keeping the pH low so harmful microbes can't take over. If this balance is disrupted from things like antibiotics, hormonal changes, sexual activity, or hygiene habits, lactobacillus levels can drop. This can lead to bacterial vaginosis, a condition where pH rises and bacteria like Gardnerella vaginalis take over. That shift can cause infection, odor, and even pregnancy complications like preterm birth. It's a clear example of how microbial balance directly affects reproductive health. Lactobacillus crispatus is like the bodyguard of the vaginal microbiome. This gram-positive rod-shaped bacterium helps keep the vaginal environment healthy by producing lactic acid, hydrogen peroxide, and bacteriocins that stop pathogens in their tracks. The big takeaway here is that microbes aren't just germs, they're our allies. By maintaining a balanced microbiota, we support digestion, immunity, and reproductive health.
Hello everyone, I will be sharing more about Bacteroides fragilis. This is a gram-negative, bacillus-shaped, though sometimes pleomorphic, obligate anaerobe capable of fermentation and anaerobic respiration and is an opportunistic pathogen. There are different strains of Bacteroides fragilis. Non-toxigenic strains are regarded as probiotic. Enterotoxigenic strains produce proteolytic enterotoxins, and other strains export proteins that are nearly identical to ubiquitin, which plays a role in immune response, and so these may contribute to autoimmune disease. It's primarily located within the colon of the human microbiota, but it can be found in other body sites as well. Some of the functions of Bacteroides fragilis include digesting complex carbohydrates that the body cannot break down on its own, and in doing so, it produces short-chain fatty acids and smaller compounds that are beneficial for other bacteria. It regulates the immune system and protects from pathogens by competing with harmful bacteria like C. diff for space and nutrients, and by secreting antimicrobial toxins that target other pathogens. Some of the harmful interactions with the host are most closely associated with the enterotoxigenic strains. These cause inflammation and tissue damage in the colon and have been linked to an increased risk of colorectal cancer. The opportunistic pathogen functions begin with the breach of the colon due to tumors, appendix rupture, surgery, injury. Bacteroides fragilis then enters extraintestinal environments and quickly progresses into infections and abscesses at multiple body sites. If left untreated, this can lead to bacteremia, which can be fatal, and bacteremia secondary to Bacteroides fragilis infections has a particularly high mortality rate of 27%. There are treatments, though few antibiotics are effective due to the enzymes that Bacteroides fragilis produces. Metronidazole is typically the first choice, and it's historically been very effective, though resistance is now becoming a concern. Okay, I believe I'm out of time. Here are my resources. Thank you so much for joining me.
Fusobacterium nucleatum is one of the most common of the core microbes in the oral cavity. It is gram-positive, anaerobic, and when it is in its most favorable environment, it can provide a significant amount of butyric acid. This is the type of energy needed for anti-inflammatory cells to do their job. When our diets are too acidic or high in sugar, it can throw off the neutral pH of the oral cavity, enabling certain opportunistic bacteria to overgrow. F. nucleatum acts as a binding agent for these other microorganisms to form dental plaque. Excessive plaque can lead to a variety of gum diseases, and if left untreated, it can become chronic or lead to more serious illness, not only in the mouth, but throughout the body. Specifically, Fusobacterium nucleatum animalis has been routinely found in colorectal tumors. You can imagine that saliva works as the gas needed for the vehicle. In this case, our make and model or family species is F. nucleatum animalis. and it can drive this harmful bacteria and pathogens from our mouth all the way to our gut. But don't fear. We have control over this balance with our diet, oral hygiene, antibiotic use, and sleep patterns. And when our internal environments are balanced, bacteria such as F. nucleatum can actually work to fight the same diseases it can cause when not balanced. This illustrates how important it is to create healthy everyday habits to keep our microbial worlds happy and working for us, not against us.
Our bodies are home to trillions of microorganisms that live on our skin, in our mouths, and throughout our gut. This community is called the human microbiome. The microbiome starts developing during birth. As we pass through the birth canal and take our first breath, we encounter thousands of microbes that begin colonizing our bodies. Throughout life, factors like diet, emotional or physical stress, disease, and medications, including antibiotics, can disrupt the natural balance of our microbiome. Let's focus in on the digestive system, specifically the lower gastrointestinal tract. The gut hosts the largest and most diverse microbial community in the body. Here, bacteria dominate, but fungi like Candida and even protozoa are present too. Clostridium difficile, otherwise known as C. diff, is one of the gut's natural microbes. It normally exists in small numbers and lives peacefully among the other bacteria. However, antibiotics can disrupt the normal balance of bacteria in the gut by killing off both the harmful and the beneficial microbes. And when the beneficial bacteria are reduced, it can create an imbalance that allows bacteria, like the opportunistic pathogen C. diff, to grow. Once the normal gut flora is suppressed, these spores germinate and multiply, producing toxins that damage the intestinal lining and cause severe diarrhea and inflammation, fever, nausea, dehydration, just to name a few. Because C. diff can form hardy endospores that survive antibiotic treatment, scientists have been exploring alternative treatments. Fecal microbiota transplantation, FMT, is a procedure used to transplant a small sample of stool from a healthy colon into a diseased one. Each healthy stool sample contains thousands of beneficial microbiota that can help improve the health of the diseased colon by repopulating it with the right kinds of bacteria. So that's a wrap on the human microbiome, the gut, and C. diff in 2 minutes. But stick around for a minute longer to hear 86-year-old Billie Iverson and some reporters talk about her C. diff experience and the miracles of FMT.
How are you going to put it? This poop transfer. That's the only way I know how to put it.
Doctors call it a fecal transplant.
Take my poop out and put somebody else's poop in there. That is the most disgusting thing. I said, oh, no way. I said, no way am I doing that.
It may sound pretty gross, but Dr. Kelly says it seems to work really well. And it's being done more and more these days to treat C. diff.
It's really almost like an organ transplant. You're taking this whole community of microorganisms from one person, you're transplanting them into another person, and then these things, take root, colonize, and kind of restore that balance.
Iverson was so desperate, she decided to try it.
I was scared to death, honey. I'm an old lady. I got one foot in the grave and nothing on a banana peel.
The procedure turned out to be really easy, and it worked pretty much overnight.
It stopped. Yeah, right away. I'm good now. I'm feeling great.
What happened?